Welcome to all in BioReside. In today's session, we will discuss about the DNA replication. In today's session, we will discuss about what is DNA replication, what are the main component of DNA replication, and replication process in eukaryotic as well as prokaryotic. Okay, so we start today's session and understand complete information about the DNA replication. Okay, so now first we understand what is a DNA replication. Okay, in DNA replication process, DNA plays a most important role. Okay, now in which form? Is DNA is a genetic material. Okay, it will transfer the information from one generation to another generation. Okay, and it will also code some information. Which types of information? To code a protein or to synthesize a protein. Okay. What is genetic? What is DNA? DNA is a genetic material. It will code the information in a form of which form and which information? To synthesize a protein. Okay. During this protein synthesis, a series of process will happen. In this series of process is known as a central dogma. And what are the main steps of central dogma? First step is a replication. What do you mean by the replication? Replication means DNA will self-replicate. Means DNA will form their own copy. Okay. After this replication, DNA is converted into the mRNA. And this process is known as a transcription. Okay. And then finally this mRNA is converted into the protein. And the process by which mRNA is converted into the protein is known as a translation. Replication, transcription and translation is known as a central dogma of molecular biology. Okay. During this central dogma, first step is a replication. Okay. In these replications, DNA will self-amplify. Means DNA will amplify into the protein, to DNA. Okay, like this. And our DNA is a double helical structure. Antiparallel strand contain the two strands. First strand is known as a template strand. And second strand is known as a complementary strand. Okay. And the direction is 5' to 3', like this, 5' to 3', and 3' to 5'. Okay. And in a case of eukaryotics, DNA is tightly packed with the help of histone protein. stone protein. During the amplification or during this replication, some modification will happen in the stone and a replication process will start. The fundamental of replication. Now we discuss about the types of replication. DNA replication. Okay, DNA replication mainly semi-conservative. Maybe semi-conservative. Semi-conservative means during the amplification process, one strand is old and second strand is new. This is means first one strand is conserved and second is new one. This is old one and this is new one. Okay, this is the semi-conservative model. Conservative and one another model is a conservative. Conservative means one strand is conserved, second strand is new one. Okay, this is the conservative. Next is a dispersive. In dispersive means some part is conserved, some part is new. Conserved, new, conserved, new. Like this. And now this is the dispersive. Okay. These are the three models. Semi-conservative, one is conserved, one is new. In conservative, one is old and another is new. In dispersive, some part is conserved, some is new. Some conserved, some new. Some conserved, some new. Okay. DNA will generally semi-conservative and the mechanism is vary. It is depend upon the structures. Your DNA may be linear, may be circular, may be nuclear, may be mitochondrial, may be chromosome, may be plasmid, may be viral. Okay. Depend upon the types, location and structures, it is divided into different model. First is your theta replication or theta model. Okay. In this theta model is seen in a double stranded DNA. And the best example is the E. coli or prokaryotic replication. Okay. Like this. DNA, circular DNA, amplification will start and form like this. One strand is old one, second is new one. Old, new, and it will amplify like in this direction. And this is in this direction. Okay. And it will convert it into the 2. This theta like structure is formed. That's why this is known as a theta replication. The production of theta replication during the 2 circular DNA. That's why this is known as a theta replication. Okay. And it will initiate with ORI region. Some helicase will bind. Then it is a bidirectional. Okay. Then same process. Then DNA polymerase will bind, lagging strand will form, leading strand, lagging strand will form, and DNA will replicate. Okay. And detail discuss, we will discuss, and the detail about this detail procedure we will discuss in our incoming slides. Okay. A special feature is intermediate look like a Greek letter, that is the theta, and this is the theta. Okay. Impedance ensure the faithful duplication of DNA of bacterial chromosome before the binary fission. Okay. Next is a rolling circular DNA or rolling circular replication. In this model, first band will nick, matlab in one strand nick will form. Okay, one strand is breakdown, then new strand will form like this. Okay, one is old and new is released one. Okay, first cleavage at this point. Now this is the 5' end, this is the 3' end, and at this 3' OH group, DNA polymerase or DNA other replication machinery will bind and start the replication process. Okay, and this old one is removed out, then finally cleavage and DNA will replicate. Okay, here no breakage, but in a rolling circular, one breakage, one DNA strand is break. Okay, new strand display old one, and old strands can become a double stranded based on a complementary. Okay, this is also a type of semi-conservative because here a one strand will conserve and one is removed out. This is also a type of semi-conservative because a one strand is conserved and second is then is new one. Okay, now linear. This linear replication process is performed in eukaryotic chromosome. In linear replication, number of ORI regions are present. Let's say ORI region 1, ORI region 2, ORI region 3. Then here on this region, replication machinery will load. Then this replication fork will happen. Then proceed like this. Then new DNA will amplify. And this is also a type of semi-conservative because one is old and one is new. Okay, these are the three main mode of replication. First is your theta replication, second rolling, and third is linear. Okay, these. Now we understand the main step of replication process, both in eukaryotic as well as prokaryotic. This replication process is divided into three parts. First is your initiation, second is your elongation, and third is your termination. At this initiation, replication process will start. At this elongation, DNA will elongate, and at this termination, process will stop. Okay, this is the replication. These are the main three steps of DNA replication. At the initiation, first and most important component is the ORI. ORI at a region, ORI at the point where the replication process will start. DNA A, DNA B, DNA C, DNA G. These are the enzymes. Okay, it will help to form a replication fork. Then SSV, that is a single binding proteins. Okay, then CDC6 and MCM. Okay, these are in a case of prokaryotic and this is for eukaryotic. These are the components which we require in initiation step. Okay, then in elongation, in DNA elongation we require DNA polymerase, DNA ligase, and SSV, that is a single binding proteins. Okay, in termination step we require telomerase enzyme in case of eukaryotic, TUS and TER protein in case of prokaryotics, and topoisomerase to remove out the positive and negative coiling. Okay, these are the most important component during the DNA replication process. Okay, now we discuss each and every point in detail. Okay, first ORI, that is the origin of replication. At this ORI, replication process will start. It will contain some specific reasons, specific sequences. Okay, and this sequences is AT rich. AT rich means AT-rich means adenine thymine rich. Okay. ORI, origin of replication, contains some specific reason. It is a starting point. Okay. In case of chromosome or plasmid, this reason is known as a ORI. That is the origin of replication. Okay. And general feature, it is a AT-rich. AT-rich means it will contain the multiple AT base pairs. Okay. And adenine bind with thymine to double bond. Okay. And this adenine bind with double bond, and this double bond is easily breakable. That's why this reason is AT-rich. Okay. Sequences means a specific recognition motif where the initiator protein will bind. Okay. In case of prokaryotes, this contain a 13-mer or 19-mer sequences. Okay. Then binding site for the initiator proteins like DNA A in a case of prokaryotic, ORC in a case of eukaryotics. Okay. Must ensure once per cycle replication to maintain the genomic stability. These are the specific, some general features of ORI. Okay. Now we discuss the difference between the ORI region of prokaryotic as well as eukaryotic. Okay. If we're talking about the number of genome, then in prokaryotics only one ORI region is present, but in case of prokaryotics multiple ORI region is present. If we're talking about the size, the ORI region contains some specific sequence and then length is 245 base pair. But in case of eukaryotics, this is the 100 base pairs. Okay. Control by the DNA A protein or sequence A, and in eukaryotic it will controlled by ORC complex and CDKS proteins. Okay. Replication timing in prokaryotics it is continuous, in eukaryotics as phase regulated. Okay. Sequence specificity, in case of prokaryotic it is highly conserved, but in case of eukaryotic it is flexible. Okay. And these are the some pointer, and these pointer are important for exam point of view. Like E. coli ORC region, it is a DNA box, it is a AT-rich. It will contain 13-mer and 9-mer sequences. Okay. In case of eukaryotic, it will contain ARS region. Okay. This ARS region, ORC will bind and ACS sequences. In case of human, multiple ORI region, epigenetic regulation. ORI ensure the bidirectional replication fork. In plasmid ORI, call even commonly used for the cloning purpose. And these are the most important pointers for the exam point of view. Okay. Now, initiator proteins. Initiator proteins means initiator protein are the first protein that recognize the ORI region and help to start the DNA replication process. Okay, then unwinding the DNA locally and the recruitment of the other proteins like helicase, primase, DNA polymerase to form a pre-replicating complex. Okay, now we discuss in case of prokaryotics. In case of prokaryotics, the initiator protein is a DNA A. This DNA A is the initiator protein. Okay. And how it will helpful? First step is the recognition of ORC region. Okay. And this ORC region, the size is 245 base pair, and this is 80 wrist. It will contain 9-mer sequences, 9-mer or 9-mer or 13-mer means number of base pair is 13 at this region and number of nucleotide, number of base pair 9 at this region. And this DNA A recognize the 9 base pairs, that is known as a 9-mer. and this is the sequences AT, A, T, CC, A, CA. Okay, at this point DNA A will bind. Okay. This region is a AT rich, that's why it will easily unwind. Okay. Okay. Now at this position, the initiator protein DNA A bind with ORI region. Let's suppose this is the region. Okay. At this position, DNA A protein will bind and it will form. Unwind this region like this. Okay. Now oligomerization. Oligomerization means multiple DNA molecules form a complex around this ORI regions. Okay. Now at this position, DNA will tilt. Why it will tilt? Because here the torsional strain. Okay. At this position, this causes the DNA bend or introduce the torsional strain. This leading the local unbinding of AT rich region. Okay. Then further recruitment of helicase. Helicase means DNA B. DNA B is a helicase enzyme. Okay. Then this DNA A recruit the DNA C, and this DNA C helpful to recruit the DNA B. And this helicase enzyme is responsible to unwind the DNA. Now this unwound the region. Okay. And this reason, the ATP dependence means DNA A require the ATP for the functional property. This is the ATP dependent reactions. Okay. And the key initiator protein is a DNA A. In short, first DNA A recognize the ORC region. In ORC region, basically it will recognize the 9-mer sequences. Okay, and it will and for performing their action, it required the ATP. Whenever this ATP will hydrolyze, due to the hydrolysis, here the unwinding process will start. Okay, a number of DNA A protein will bind. It will create the torsional state. Due to this torsional state, this the hydrogen bond between the adenosine and triphos in adenine and thymine, the hydrogen bond between the adenine and thymine is break down. Whenever this bond is break down, then DNA will unbound at this position. Whenever this DNA is unbound, then DNA A recruit the DNA C, then DNA C recruit the DNA B, and this DNA B is a type of helicase. Helicase is a process, helicase is an enzyme, it will helpful to unwind the DNA. Okay, this is the initiation process of prokaryotes, and at this initiation process, DNA A is a initiator protein and it is the signature protein of this step. Okay. Now, initiator protein in eukaryotics. Okay. The eukaryotic replication process is more complex as compared to prokaryotics. Okay. In eukaryotics contain the multiple ORI region. It will contain multiple ORI region. Okay. And this regulation or this DNA replication process is compact and tightly regulated by the cell cycle. Basically in G1 phase or in S phase. Okay. In G1 phase, initiation will start, but in G1 phase, this initiator protein will inactive. At this S phase, it will convert into the active form, and replication process will start. Okay. G1 phase means preparation state, and S phase is a synthesis phase. Okay. Here ORC protein plays a most important role, ORC1 to ORC6. ORC1 and ORC region recognize the ARS region. ARS region is a, what is this? This is a origin of replication point, meant at this point replication process will start. Okay, and here this ORC protein is similar to the DNA A, and DNA A is an initiator protein of prokaryotes. Okay, and this ORC will bind at a G1 phase. Okay, after this it will recruit the CDC6 and CDT1. Okay, it will bind. Okay, and this is then MCM protein will bind. What is this MCM helicase? It is a type of helicase protein. It will helpful to unwind the DNA. MCM means mini chromosomal maintenance protein. Okay, in G1 phase this MCM is in an inactive form. Okay, whenever the cell is go to the S phase, then kinase means CDKK or CDC and DDK will bind. In G1 phase this complex in a form of inactivate, but in S phase some kinase protein will bind and convert it into the active form. Okay, and in S phase initiation process will start. Okay, now what is the key differences? Key differences is that in prokaryotes main initiator protein is a DNA A, here ORC regions. Here the DNA region recognition ORC, here multiple. Helper protein is a CDC-C, here the helper protein is CDC6 or CDT1. Helicase recruit, that is a DNA V helicase, here MCM complex. Regulated by the ATP hydrolysis, it is regulated by the cell cycle kinase, that is the CDKK and DDK. It will helpful to activate the initiator complex. Number of region is only one, and in eukaryotes multiple. Okay, these are the key differences or comparison of initiators in eukaryotes as well as in prokaryotes. Now the exam point 2, DNA is a prokaryotic initiator protein, ORC is a eukaryotic initiator protein, DNA-C is the helper protein. In prokaryotes CDC6 and CDT is a helper protein in case of eukaryotes. And in prokaryotes this whole process is controlled by the ATP hydrolysis in case of protein. In case of prokaryotic cells or in case of eukaryotic cells, this complex is maintained by the cell cycle kinase, that is the CDKK and DDK. Okay. Now next is the helicase. This helicase is the enzyme. It will helpful to unwound the DNA. Okay. It will responsible to unwinding the DNA. Helicase is the ATP dependent molecular motor that move directionally along the nucleic acid strand to separate out the two strands while breaking the hydrogen bond between the base pairs. Okay, if we're talking about the types of enzyme, it is a motor protein and it is a type of ATPase because it will require the ATP to perform their task. Then energy sources is a hydrolysis of ATP in ADP. Okay, action, it will unwind the DNA, means it will break down the double stranded, it will convert the double stranded DNA into the single stranded by breaking the hydrogen bond between the nitrogenous base. Okay, if we talking about the directionality, the directionality is 5' to 3' and 3' to 5'. Okay, now in case of prokaryotics, helicase protein is a DNA B. In case of eukaryotics, helicase protein is a MCM complex. Okay, if we're talking about the structure, in case of prokaryotic, it is a hexameric protein. Okay, CDC-C and CDV-B helpful to load the DNA B proteins. Okay, if we're talking about the polarity, the move 5 prime to 3 prime on a lagging stand, but 3 prime to 5 prime on leading stand. Functional, it will unwind. Okay. In case of eukaryotic structure, it is also a hexameric protein. CDC 45 and GINS complex will helpful to recruit the MCM proteins. Okay. Polarity 3 prime to 5 prime in leading stand. Open to convert the double stranded DNA into the single stranded DNA. Okay. And also coordinate the DNA polymerase enzyme. Okay. Some pointer, prokaryotics, helicase 5 prime to 3 prime at lagging stand. MCM complex 3 prime to 5 prime activated by the CNG in leading stand. And processes depend upon the energy, basically ATP hydrolysis. It is a hexameric space structures. Helicase is a part of Replication. Okay. Mutation, it will form the genetic instability and diseases. Next, we discuss about the single stranded binding proteins. Means single stranded binding proteins are the DNA binding protein that bind the single stranded DNA region during the replication process, recombination and repairing. It will helpful to prevent the secondary structures or degraded by the nucleus. Means helicase enzyme will unwound the DNA, then this SSP protein will bind, it will helpful or it will prevent the renaturation process. Okay, helicase will denature the DNA and this SSP protein will maintain the denaturations, and it will also helpful to preserve or to prevent or to save our DNA from nuclease enzyme. Okay. In case of prokaryotic, this is the SSB protein. It is a tetrameric structures and molecular weight is a 19 kilodalton. In case of eukaryotic, it is a RPA protein. It is also a heterotrimeric proteins. Next, function is stabilize the DNA, means prevent the reannealing, keep the DNA extent in linear conformations, prevent the secondary structure, protect from the nuclease enzyme, facilitate the binding of other proteins like DNA polymerase, like primase. Okay. Work with DNA polymerase during the Okazaki fragments. Okay, and it will also helpful in DNA repairing and recombination. Now we discuss about the topoisomerase. What is the topoisomerase? Topoisomerase is an enzyme that will helpful to prevent the or to break the positive supercoiling. Okay, DNA is a double helical structure that must be unwound during the replication process. During the unwinding by the helicase, this helicase generate the positive supercoiling in a replication force or torsional stress. Okay, without correction, replication would stall. Okay, topoisomerase is the enzyme that resolve this supercoiling. Okay, by breaking or rejoining the strand. Okay, there are two types of topoisomerase: type 1 topoisomerase or type 2 topoisomerase. This type 1 topoisomerase generate the cut only one strand. Okay, it does not require any types of ATP, and functional, it will release the torsal stress. Okay, mechanism, cleave only one strand, uncut strand pass through the neck and relax the tension. Okay. And in case of prokaryotics, the example of type 1 is a type topoisomerase 1 or third. In case of eukaryotics, the example is a topoisomerase 1. Next is a type 2 topoisomerase. It will generate the cut in both the strand. Okay, both strands are unwound and rejoined. Okay, it will require the ATPs for the recruitment. It will release the positive supercoiling. Okay, mechanism same. It will generate the cut into both strands, then duplex pass through the break and realienate by the DNA ligase enzyme. Okay. In case of prokaryotics, this process will perform by the DNA guidase or topoisomerase 4. In case of eukaryotic, it process will perform by the topoisomerase alpha and topoisomerase beta, or second alpha or second beta. Okay. Now we discuss about the next exam, that is the primase. What is the role of primase? The primase recruit the RNA sequences. It will add 10 to 12 RNA sequences, and it will also produce the 3' OH group, because DNA polymerase require the free 3' OH group to perform their activity. Okay. Location of replication: primer act as a replication fork where the DNA unbound the helicase. Okay. It work in coordination with helicase to form a complex like primosome. Primase in prokaryotics: E. coli, primase is encoded by the DNA G. Work along with DNA B. Okay. It will also helpful to recruit the DNA polymerase third and extend the replication process. In case of eukaryotics, primase is a part of In case of eukaryotic, primase is a part of multiple subunit complex. Okay. DNA polymerase alpha, it is a primase complex, and the structure, it will contain the 2 primase subunit, small and large, and this is recruit the 10 RNA primers. In DNA polymerase alpha subunit, it will extend with primase by 20 to 30 deoxyribose nucleotide proteins. Okay. After the primer switching, DNA polymerase delta is bind on the lagging strand, and DNA polymerase epsilon bind to the leading strand and perform the elongation. Now we discuss about the polymerase enzyme. DNA polymerase is the enzyme that is responsible for synthesize the DNA strand during the DNA replication process, and it will also helpful in DNA repairing machinery. Okay, and this will add the nucleotide sequences at the 5' to 3' direction. Okay, during the template strand, it will require the free OH group. General feature: direction is the 5' to 3'. Template strand require the DNA template. It is a template dependent DNA polymerase, because it means it need to DNA template to synthesize the new strand. Primer requirement, because it will require the 3' for the recruitment. It will require 3' OH group, free 3' OH group. Proofreading activity is a 3' to 5' exonuclease activity. Catalytic activity means polymerization occurs by the phosphodiester bond formation. Okay. Now these are the DNA polymerase enzyme in prokaryotics. DNA polymerase 1, 2, 3rd, 4th, 5th. DNA polymerase 1 function 5' to 3'. Addition of nucleotide 3' to 5', exonuclease activity 5' to 3', also exonuclease activity. Okay. Role: removal of RNA primer, replaced with DNA sequences. Not meant replicated polymerase, but essential for the Okazaki fragment and DNA repairing. DNA polymerase second, it will also helpful in DNA repairing machinery. It will contain 3' to 5' exonuclease activity. Okay. Next is a DNA polymerase third. It is a most and main enzyme, main DNA polymerase, main replicative enzyme. Processability is very high. It will contain the 3 subunits. First is the alpha, epsilon and theta. Alpha is for the polymerization, epsilon is for the proofreading, and theta is for the stabilization. It will also synthesize both leading strand as well as lagging strand. Okay. Polymerase 5th and polymerase 5, it is a member of Y family polymerase. Okay, it will helpful in DNA repairing machinery. Okay, in SOS repairing. Okay, it lack the proofreading activity. It will introduce the mutations. Okay. Now these are the differences. DNA polymerase 1 is the primer removal or DNA repairing. Proofreading activity, yes. Unique feature, 5' to 3' exonuclease. DNA polymerase second, DNA repairing, proofreading activity, yes, but error corrections. DNA third, main replication enzyme, yes, high processability. DNA fifth and 5, SOS repairing, no proofreading activity, error prone or translation sensitive. DNA polymerase in eukaryotes. Okay. Major replicating polymerase enzyme is a DNA alpha, has the primase activity, synthesize the short RNA or DNA primers. Okay. But processability is very low. DNA delta, mainly enzyme for the lagging strand. Okay. High processability with PCNC slider ring. Okay. Also contain 3' to 5' proofreading activity. DNA epsilon, it is a main enzyme for the elongation process in eukaryotes. It will elongate the DNA or synthesize the leading strand. High fidelity, proofreading activity, and main enzyme for the replication process. Other is a beta, epsilon, eta, iota, and kappa, and zeta and theta. These are the other DNA polymerase enzyme. Now exam pointer. Kornberg discovered the polymerase 1 enzyme, polymerase first. The polymerase third is a main replication enzyme for in eukaryote. Sorry. Now exam pointer. Kornberg discovered the DNA polymerase first. DNA polymerase third is a main replicating enzyme in bacteria or prokaryotes. Epsilon is for the primase. Delta is for the lagging, epsilon is for the leading in case of eukaryotes. In case of eukaryotes, replication process is conducted by 3 types of DNA polymerase, that is the alpha, epsilon or theta. Okay. Epsilon beta is for the DNA repairing, epsilon gamma is for the mitochondrial replication. Okay. For the primer removal, we require the polymerase 1 or RNase H or FEN1 in case of eukaryotes. Okay. TLC polymerase like eta, neta, ota or kappa. It is a high fidelity, allow the bypass of lesion. These are the exam pointer.
Next is a slider clump. Slider clump, it is a processability factor in case of DNA polymerase. Processability factor means it is a type of slider clump. It is a ring-like structure protein. Okay. It will helpful to DNA polymerase. It will anchor the DNA polymerase to temperate strand. Okay. To increases the processability of DNA polymerase. Processability of DNA polymerase means DNA can synthesize the long strand of DNA without any types of dissociation or any types of disturbance. Okay. If we're talking about the structure, this is a ring-like structures. In case of prokaryotes, it is a beta clump. In case of eukaryote, it is a PCNA. Now, next is a clump loader. What is a clump loader? A clump loader is a multiple protein. It will helpful to recruit or to load the slider clump. Okay. It belongs the AP plus or ATPs family. It means ATPs associated with diverse cellular activities. What is the function? It will need to slider clump, beta clump in case of eukaryotic and PCNC in case of eukaryotics. Cannot spontaneously open in circle DNA. Okay. This slider loader use the ATP hydrolysis, open the clump and open its primer template junctions where the DNA polymerase can then bind and begin the elongation process. Basically, this is a most important for the elongation process. Okay. Now, Okazaki fragment. What is the Okazaki fragment? This Okazaki fragment is a stand of lagging strand. Okay. The direction of DNA polymerase is 5' to 3', but the direction of lagging strand is a 3' to 5'. Okay. DNA polymerase is a semi-conservative and bidirectional. Okay. DNA. Now, what is the Okazaki fragment? Okazaki fragment is formed in lagging strand. It is a small fragments. Okay. In case of prokaryote, it is a 1000 to 2000 long. In case of eukaryote, it is a 100 to 200 base pairs. Okay. The direction of lagging strand is 3' to 5'. Okay. How this fragment will form, how the complete process we will discuss in our incoming slides. Okay. Function, helicase unwinding, produce the single standard template, primer is required. 8 primers, add only 10 nucleotides. DNA polymerase third in case of prokaryotes, in polymerase DNA delta in eukaryotes, extend the fragment from primer, synthesize the DNA 5' to 3'. Protein involved helicase to unwinding, SSP for preventing the DNA link, primase to add the RNA. Now we discuss the key protein that are involved in a formation of Okazaki fragment. First is the helicase to unwind the DNA, SSP to prevent the DNA link, primase to synthesize the RNA primers, DNA polymerase third or delta for the elongation process, DNA polymerase 1 or RNase H or FNP1 to remove out the primase protein, DNA ligase to join the fragments, slider clamp and clump loader to increase the processability. Okay. Exam point of view. Okazaki fragments are on a lagging stand. Length differ in case of eukaryotes or prokaryotes. Enzyme prokaryotes DNA polymerase 1 is required. In case of eukaryotic, RNase H or FEN1 required. DNA ligase required the ATP in case of eukaryotics and FED plus in case of prokaryotics. And the discontinuous replication model proved by the Okazaki experiment using the phase cheese labeling with radioactive nucleotides. Now, primer removal and gap. Next is the RNA primer removal and gap filling. Okay. During the replication process, in Okazaki fragments, small fragments are formed, and this RNA primer must be removed or replaced by the DNA sequences and then ligate by the ligase enzyme. Okay. Problem: DNA polymerase cannot start the de novo synthesis. That's why we require the primer enzymes. Okay. And this RNA primers or RNA sequence must be removed out. In case of eukaryotic, this process will performed by the DNA polymerase first. Okay. It contains the 5' to 3' exonuclease activity, 3' to 5' exonuclease activity, and 5' to 3' polymerase activity. Okay. And this process is called as a Nick translation. Okay. In case of eukaryotic, this process is performed by the delta. It will synthesize the Okazaki fragment. Okay. Flap endonuclease 1 and RNase H are also helpful to remove out the RNA primer. RNase H degrade the RNA primer and flip endonuclease remove the last nucleotides by flipping process. Okay. Then DNA polymerase delta can extend the DNA sequence, extend the DNA and fill the gap. Okay. And this gap is filling by in case of prokaryote, the gap is filling by the DNA polymerase 1, and in case of eukaryote, this gap is filling by the DNA polymerase delta. And this nick is filling by the DNA ligase. What is the DNA ligase enzyme? This DNA ligase is a very essential enzyme. It will join the DNA fragment by forming the phosphodiester bond. Okay, between the 3' hydroxyl group or 5' phosphate group. Okay, and it will also play the most important role in DNA replication, in DNA repairing and recombination process. Role on leading strand, replication continue, so the ligase is really required. But in lagging process, replication is discontinuous. That's why Okazaki fragments are formed. These Okazaki fragments are joined by the DNA ligase enzyme. Next is a telomerase enzyme. Okay, this telomerase enzyme is helpful to terminate the replication process in eukaryotes. Okay, telomerase is a ribonucleoprotein enzyme complex. It is responsible to maintain the length of telomerase. Okay, telomerase are the repeated sequences. Like in case of humans, it is the ATA, TT, AGGG. Okay, it will helpful to protect the chromosome at the end of the point during the fusion process. Telomerase enzyme is the most important protein in eukaryotic termination process. Okay, this is the termination process proteins in case of prokaryotics. Okay, in case of prokaryotics, ter protein and tus proteins are most important. Okay, ter protein is a termination process. It will specific 23 nucleotide base pairs. Okay, located at the replication termination point. Okay, ter protein function is the polarized manner. Okay, replication fork can pass from the permissive site. Replication fork is not passed through the non-permissive site. Thus tus protein is bound. This protein is permissive and this side is non-permissive. DNA helicase cannot pass this site. Okay, that's why this process will terminate it. Around the 10-ter site, that is a ter A to ter J, but not all are equally functions. Most effective are ter protein A, B, C, D, E, F, G. Okay. What is the help? It will help the replicant fork trapping, ensure the replication fork meet the control termination zone. Okay. Next is the tus protein. Tus protein is a DNA binding protein. In case of prokaryotic or E. coli, especially recognize and bind to the ter protein. And that molecular weight is at 36 kilodalton and binding slightly to ter protein. And it is from the tus to ter complex. Okay. Tus to ter complex form the tus and ter complex. And the DNA helicase cannot pass this tus or ter complex. That's why this region converted into the non-permissive region and replication process will stop. Thank you so much everyone. Please like, share and subscribe. Why or is I?